Hello everyone, this is Suchi. Welcome to my channel. If you are watching my video for the first time, subscribe and support and click on the bell to get the notifications of my new videos. In our previous video, we learned one of the process of cell division, that is mitosis. Today, let's know about the other process of cell division, meiosis. As you already know, sexual reproduction in organisms takes place through the fusion of male and female gametes, that is, the sperm and egg, respectively. Gametes are haploid in nature, that is, they contain only half the number of chromosomes. This genetic content makes them different from other body cells. Meiosis leads to the formation of haploid cells. As you already know, mitotic cell division is equational in nature, while meiosis is reductional in nature. This meiosis occurs in two stages: nuclear and cellular division, that is, meiosis 1 and meiosis 2. DNA replication occurs only once. It involves the pairing of homologous chromosomes and recombination between them. Four haploid daughter cells are produced. Here, diploid cell is reduced to two haploid cells. We can see four stages in meiosis, that is prophase 1, metaphase 1, anaphase 1 and telophase 1. Now let's know about prophase 1. This is the longer than mitotic phase. Further, this is divided into five substages: lactotine, zygotine, pachytine, diplotine and diakinesis. Let's know what these substages. In lactotine, chromosomes begin to condense and attain compact structure. In zygotine, the pairing of homologous chromosomes start, a process known as chromosomal synapses. A pair of synapsed homologous chromosomes forms a complex known as bivalent or a tetrad. At Pachytine stage, crossing over of non-sister chromatids of homologous chromosomes occurs at the recombination nodules. The chromosomes remain linked at the sites of crossing over. Diplotid stage shows the separation of homologous chromosomes of the bivalent exerts at the sites of crossover. The X-shaped structures formed during separation are known as chiasmata. Now, the diakinesis shows the termination of chiasmata and assembly of the meiotic spindle to separate the homologous chromosomes. Next, nucleolus disappear and the nuclear membrane breaks down. These were the changes that occurred in the first stage, that is prophase 1. Now let's know about metaphase 1. The bivalents align at the equatorial plate and microtubules from the opposite poles attach to the pair of homologous chromosomes. In the next stage, that is anaphase 1, the two chromosomes of the each bivalent separate and move to the opposite ends of the cells. The sister chromatids are attached to each other. In telophase 1, the nuclear membrane reappears and is followed by cytokinesis. Now let's know about meiosis 2. Even meiosis 2 has four stages, same as meiosis 1: prophase 2, metaphase 2, anaphase 2 and telophase 2. The meiosis 2 is same as mitotic cell division. So let's know about the changes that occur in prophase 2. In prophase 2, This stage starts immediately after cytokinesis when the daughter cells are formed. Here the chromosomes condense, the nuclear membrane dissolute, and the cell organelles disappear. Metaphase 2, the chromosomes connect to the centrioles, poles, and the kinetochores of sister chromatids through microtubules, and they align at the equator to form metaphase plate. Next, anaphase 2. There is splitting of centromere and the sister chromatids are pulled away towards the opposite poles. Next, telophase 2. Here the chromosomes dissolve again, nuclear envelope develops around it, and cytokinesis occur. As a result, four haploid daughter cells are formed. So this was about the cell division by meiosis. Now let's know about the significance of meiosis. Here, reproduction in animals takes place through the fusion of gametes, that is, two cells fuse together with their genetic material to develop a zygote. If the germ cells which give rise to gametes also contain chromosomes like during mitotic cell division, like in somatic cells, the zygote will have an accumulation of chromosomes in its nucleus. This accumulation will keep on increasing with every subsequent generation. So, meiosis offers a very smart solution by reducing the number of chromosomes in the gametes to help of their parent germ cell. Also, prophase 1 of meiosis allows recombination of homologous chromosomes. This is essential for the variation to be introduced in the genetic makeup of gametes in the new generation, which is responsible for evolution. Now let's have some glance about the key points for meiosis. Meiosis is a reductional process in which a single cell divides twice to produce four cells with half the original amount of chromosomes. The number of chromosomes is reduced to half, that is, from diploid to haploid. Crossing over is a process of meiosis in which two chromosomes of a homologous pair are exchanged between non sister chromatids. So, in meiosis 1, homologous chromosomes separate from each other, two diploid daughter cells are produced, and in meiosis 2, sister chromatids separate and four haploid daughter cells are produced. So, this was all about the meiosis and the importance of meiosis. Hope this was helpful, and let's stop here. This was about the cell division. If any queries or requests, please drop in the comment section. Subscribe, support, and click on the bell to get notified. Thanks for watching.